Bismillahirrahmanirrahim. Assalamu alaikum. Dear students, today we are going to study the new topic, that is the immunity, resistance of the body to the infection, which is called immunity. First of all, I would like to define the immunity. It is the ability to resist infection and disease. Previously, we have studied the process of inflammation, how it occurs, what are its signs and symptoms, and how the neutrophil and macrophages they help to defend our body against these infective agents. If we go in further detail of the defensive action of our body, we come to know that many other cells and organs they are playing a very important role regarding the body defense against infective agents. Many body cells and tissues are involved in the implementation of the immunity. Mainly we can say that some of this immunity is natural and some we acquired later on with the exposure of any harmful infective agent. So we can say that some of the immunity is by birth. It may be called as innate, non-specific, natural, all these names for one type or the first type of the immunity. It can involve the epithelium on the body surface or occur in connective tissue. In the GIT system, or it may involve a cellular response. The second type is specific, acquired, or adaptive. This specific type of the immunity involves the production of lymphocytes, either the B lymphocytes or the T lymphocytes. But you must appreciate this point that these two types they are going with each other. They are going together with each other, or we can say they are working parallelly. As I have told already that the inflammation process is just to defend against the harmful agent. If it is subside by the neutrophils, macrophages, the further cells or the further processes cannot be initiated. The process of defense of the body that is going on, that continue unless and until the complete destruction of the infective agent. This defensive function of our body, it involves many types of the cells, organs or molecules. Regarding organs, we can have tonsils, adenoids, thymus, lymph nodes, spleen, fire patches, appendix, lymphatic vessels, bone marrow. All these cells, they are lined by macrophages or producing a specific type of the cells which are defensive in function. Regarding the cells, we can have lymphocytes, B lymphocyte or T lymphocyte, plasma cell, natural killer lymphocytes. With them, we can have monocytes, macrophages, and granulocytes, as we have already discussed the role of the neutrophil, eosinophil, and the basophil. You can see from this picture the view of different defensive cells like natural killer cells, eosinophils, neutrophil, basophil mast cells, and the monocyte macrophages. Different type of the molecules involving antibodies, complement systems, cytokines, interleukin, interferons, all these also have an important role regarding the immune system of our body. After the definition of the immunity, we are going to classify it. This classification is not written as such in your book, so you must copy it, and whenever it is asked from you to classify the immunity, you must tell this classification, which according to this classification, the main immunity is divided mainly into two categories: innate or natural, adaptive or acquired immunity. This acquired adaptive immunity is further subdivided into natural and artificial. Each one is further subdivided into passive and active. Passive natural immunity is through the mother's milk or the things or special types of the cells which cross placenta coming from the mother to the newborn. An active infection that involves the humoral and cell mediated. Artificial involve the antibody transfer and the active immunization against specific diseases. We can have another classification also. This classification is very simple, easy to remember. If you go in somewhat more detail, you can have this type of the classification. Again, the main categories are innate and adaptive immunity. These things that are the skin, mucosal membrane, they are the natural. And specific, then we can have non-specific internal defenses like these natural cells, macrophages, interferon, complement and inflammation. Through these process, you can have the immunity. You can get the immunity with the exposure of or with these developments. Then adaptive immunity involves T lymphocyte, B lymphocyte and the plasma cells. As far as the characteristics of these two are concerned, this is a very nice tabulated form to describe the characteristics of innate and adaptive type of the immunity, and at the same time we can compare these two types. So again, this is not present as such in Guyton, and you must concentrate to this slide also. Specificity: in the innate, we have the structures shared by the groups of related microbes, while in adaptive, for antigens of microbes and for non-microbial antigens. Diversity: it is also there. Memory: in the innate, we have no memory. while in the acquired we have there, and so on. Again, I am describing these things in very simple words to clearly differentiate between the innate and acquired immunity and other words used the previous during the description of the previous slides. Innate, that is present at birth, acquired, develop after birth. Active immunity is antibodies develop after exposure to antigen, while the passive immunity is antibodies are transferred, that is antibodies, they are transferred from one another source. They are either naturally acquired antibodies that are coming from the mother through the placenta, or by an injection of antibodies made in an other animal. Naturally acquired, that is through environmental exposure to the pathogens and induced through vaccines containing pathogens. This is almost the summary of the topic of the immunity. Now we will discuss one by one in more detail how they occur, how they work. Regarding the first line of defense and the things which are concerned with the innate immunity, these are mainly the skin and mucosal barriers. Then the second line of defense, that is played by neutrophils and macrophages. Immunologically, natural killer cells destroy abnormal cells. Interferons, they are the chemical messengers that coordinate the defenses against viral infections. Complement an inflammatory process, and the fever, high body temperature which increases body metabolism, accelerates defenses and accelerates body defenses. For the external defenses, the skin and mucous membranes, it must be intact. If there is any injury to this barrier, the infective agent like bacteria or virus can easily go inside the body. So, to defend these microbes, the skin and mucous membrane should be intact. Certain cells of the mucous membranes produce mucus that traps microbes and other particles, for example, nasal cavity in the trachea, ciliated epithelial cells in the lungs. Then we can have the process of phagocytosis through the different phagocytes. They ingest the invading microbes and initiate the inflammatory process. Remember it that all these processes of body defense, they are going together or they are going parallelly. Again, the assistance played by complement system, natural killer cells, lysosome, as I am telling you again and again, the role of these things, they are going together. If the infective agent is simple and its strength is low, it's very easy to kill it. So that or it's very simple to kill it, so that very simple or the simplest mechanism of killing these agents that are there. More and more processes they become involved against these agents. If their strength is strong, the number of infected agent is much more, or the injury is more as compared to the simple injury that is involving just few tissues. After that introduction of the innate immunity, we come to the second type, acquired or adaptive immunity. Innate immunity does not require much more details because we have already studied the inflammatory process, we know the barrier of the skin and all these, and also the gastric juice, that is HCl, produced in stomach. It is also a type of the barrier. With that, we switch to the acquired immunity, which mainly involves the lymphocytes. This type of immunity is the major kind of defense and provide extreme degree of protection. Mainly it is of two types, humoral and then cell mediated. Humoral or B cell immunity involving the secretion of antibodies, and then T cell immunity which involves the activated T cells. Either we have the innate or the acquired immunity. We are again and again saying that immunity is the defensive process against the infective agent. The agent which is going to give any harm to our body. For that purpose, we must know the definition of antigen. As many students say that antigen is or the immunity is against the foreign particle which is going to produce any harm to the body. For that, any bullet which is going to give the harm to your body, that may be called as antigen. So for that purpose, we must be clear about the definition of antigen. What is the definition, exact definition of antigen, so that we must know that this type of the thing that is going to produce the immunity process, that is antigen, and that is going to produce the antigen antibody reaction. So what is the definition of antigen? These are the protein or large polysaccharides, and it is they that initiate the acquired immunity. This is a very specific substance with the high molecular weight 8000 or greater and having the regularly recurring molecular group which is called the epitopes. So this is the exact definition of antigen. These are the specific antigenic substance with the high molecular weight and the specific group, special group epitopes, they are regularly, repeatedly there. So any substance which fulfills this criteria, we name it as antigen. And then this is the substance which is going to produce the antibody reaction. A lymphocyte actually recognizes and binds to just a small accessible recurring portion of the molecule on antigen, which I have already discussed, that is called the epitope. And that makes antigen very specific. So the lymphocyte will attach to this specific molecular group, and then the antigen antibody reaction starts. As we all know that in our body, lymphocytes are present mainly in the lymph nodes and also some other lymphoid tissues located at throat, pharynx, GIT, spleen, thymus and other areas. They are mainly derived from the pluripotent hematopoietic stem cell, and from these cells you can see here, from these cells two specific type of the lymphocytes they grow, B cell and T cell. T cell, some of these cells that are going to thymus, because of that they are named as T cells, and some of these cells they are initially in the fetal life develop into liver and then the bone marrow, that are going to be B lymphocyte. From the thymus, T lymphocyte they are formed, and in this way the B lymphocyte they are formed through the T lymphocyte. Cell mediated immunity is initiated, while through the B lymphocyte the humoral type of the immunity that is initiated. From there the B cell or either the T cell they come to the blood, lymph and lymphoid tissues. Pre-processing of T cells and the B cells. That is very important. Once these cells they are formed, they come to the thymus. Here one thymic lymphocyte develops specificity against one antigen and another against another. These cells then leave thymus and spread to all lymphoid tissues of the body through the blood. So it is very important to know that from the stem cell, some of the cells they are going to the thymus where their pre-processing occur. What happens? In the thymus, they are getting the training. These cells, they are trained in such a way that they can differentiate between the body tissue cells and the foreign tissue cells. And here, the specificity and diversity is there, that all these cells they come in contact with the normal body own tissue cells, and they are trained to differentiate between these cells and the foreign cells. So this pre-processing is very important. If there is any fault occur during this process of pre-processing of T lymphocytes during their stay at thymus, that can develop the disease process. In many autoimmune diseases, these cells they are unable to differentiate between the body's own specific cells and the foreign cells. so that they may injure the body's own cells that create the autoimmune diseases. For the, for the human immunity, the specific cells they are coming to the fetal liver and then to the bone marrow. B lymphocytes, they are named as such because this population of cells was first discovered in birds, which have a special processing organ called the bursa or fabricans. For this reason, they are named as B lymphocytes. B lymphocytes are different from T lymphocytes in two ways mainly. Instead of the whole cell developing reactivity against the antigen, as occurs for the T lymphocytes, B lymphocytes actively secretes antibodies that are the reactive agents. These agents are large protein molecules that are capable of combining with and destroying the antigenic substance. So, as from the previous pictures of diagram, you come to know that from the stem cells, some of the specific lymphocytes, they come to the thymus. They are named as T lymphocytes. Whenever they come to the through the lymph and through the blood, they come to the tissue cells or the lymphoid cell, specific lymphoid cells. Whenever any antigen come in contact with them, they become much more activated or they are changed to their activated form. As far as the B lymphocytes they are concerned, they are going to produce the specific antibodies. The second difference between the B lymphocyte and the T lymphocyte is that B lymphocytes have even greater diversity than the T lymphocytes, thus forming many millions of types of B lymphocyte antibodies with different specific reactivities. After pre-processing, the B lymphocyte, like the T lymphocyte, migrate to lymphoid tissue throughout the body, where they lodge near but slightly removed from the T lymphocyte areas. What happens when specific antigens come in contact with T and B lymphocyte in the lymphoid tissue? Certain T lymphocytes become activated to form the activated T cells, while the B lymphocytes they become activated and form the antibodies. But you must remember that these processes they are very much specific. For the specific antigen, we have the specific antibodies to be reacted, and the antigen antibody reaction is very much specific. All the different lymphocytes that are capable of forming one specific antibody or T cell, they are grouped together and called the clone of the lymphocytes. Lymphocytes in one clone, they resemble with each other. Through this, I want to show you how the specificity regarding antigen antibody reaction is there through the B lymphocytes. You can see these are the different types of the B lymphocytes. Very specific antibodies, they are secreted through specific B lymphocytes. For example, in simple words, any antigen which will be triangular and shape that is going to be attached with this one. Only the circular type of the antigen that will be attached to this one. Only rectangular type of the antigen that will be attached to this type of the antigen, that is this type of the B lymphocyte. In any antigen antibody reaction, when the specific type of the antigen attached to the B lymphocytes or come in contact with the B lymphocyte, it is going to be attached with the specific and B lymphocyte antigen. Other, they will not react with this one. Once it is recognized that this type is going to have the antigen antibody reaction, now it must be increased in number to kill completely the infective agent. So this specific type of B lymphocyte then proliferate or divide into many other B lymphocytes of the same specific type. Others will rest, but this type that increases much more in number, so that at a greater rate the antigen antibody reaction that take place. Mechanism of activation of clones. Antibodies present on B lymphocytes act as BCR. IgM will activate antibody producing machinery when comes in contact with antigen to produce antibodies, so on, and I have told you already. Through this animation, I want to show you how the B lymphocytes have antibodies over them and how it reacts with antigen. This is the antibodies on the surface of the B lymphocyte. Antigen coming to be attached with the surface of these antibodies. B cell receptors bind to specific intact antigens are often called membrane antibodies or membrane immunoglobulins. This process I have already told you that the specific antigen is coming in contact with the specific antibodies only, not to each other. But at this stage, I must tell you one more thing, that some of the specific T cells, they are also going to assist B cells. These specific type of the T cells, previously we have studied the T cells that are activated whenever come in contact with the antigen, and that is called the cell mediated immunity. But in this picture or in this figure, I am going to describe the help of the specific T cell to the B cells. That is, they are also assisting the B cell type of the immunity at this stage, that when they need the attachment of specific antigen to antibody, specific T cells, they help them, and they are named as helper T cell, so that it becomes easy for for B cell antigens to be attached with the antibodies, B cell antibodies to be attached with the antigen for the antigen antibody reaction. Because of the assistance of helper T cell, specific type of B cells, they release antibodies in much more greater number as compared without their assistance. So at this stage, The specific T cells, which are named as helper T cells, they are assisting this process, or B cells, the process through which the antigen-antibody reaction takes place, and as a result of the assistance of the helper T cell, much more antibodies they are released by the B cells. The process of immunity that will be continued in the next lectures, and we will discuss this antigen antibody reaction. Firstly, the structure of the antigen, how it is going to be to react with antibodies, and what is the complement system, and all other things. So keep watching these lectures. Now I wish that you have a good knowledge about the antigen antibody reaction. So thanks, and Allah Hafiz.